Sure. Sorry. Go for
Sure.
it.
Okay. Can the sunflower sea star aid the recovery of kelp forests? So, over the past 4 or 5 years, we have come to appreciate, because of their absence, the role played by sunflower sea stars in maintaining kelp forests by devouring purple sea urchins. The average sunflower sea star can munch through about 5 purple sea urchins in a week, and they don't seem to be picky about the quality of their food. And that's good news and valuable data for efforts to reintroduce these now endangered predators to coastal ecosystems along the west coast of North America. It suggests that if sunflower sea stars return to their former habitats, they could help to keep purple urchin populations in check and possibly help restore kelp forests. Aaron Galloway, our next speaker, is an associate professor at the University of Oregon's Institute of Marine Biology, the Marine Lab near the mouth of Coos Bay. He leads the Coastal Trophic Ecology Lab, which is generally focused on trophic inferences in marine benthic food webs, with an emphasis on algae, invertebrate interactions, and kelp forest ecology. So he's going to tell us about a project of which he is co-lead, along with Sarah Gravem at Oregon State University, to better understand the role of sunflower sea stars in sustaining kelp ecology. And Aaron, I am all ears, and take it away. Maybe he's on mute.
Okay, yeah. I think I'm in the clear now. Can you all hear me?
Yes.
We can hear you. Thank you.
Great. And can you see the main screen, or do you see the?
Yep.
All systems go.
And you can see me as well.
Yes.
All right, cool. Thanks for the introduction. Thanks for inviting me to attend this conference. I've attended this conference in the past and enjoyed it. So it'll be fun to offer a different perspective. I think my talk has not a single photograph. Well, shoot, there is one photograph of an otter. Dang it. Anyway, it's going to be a different perspective, although I'll put it in context a little bit with sea otters. And so you've already given the introduction, so I'll get right to my first. See here, make sure I can advance slide, which is, as you know, science is a team sport, and any scientific process that we undertake, it involves the team. And I'm really excited to talk about our teamwork today. The PIs that are involved are myself, Dr. Sarah Gravem, and Professor Chrissy Croker. And the work that I'm going to talk about largely is under the umbrella of our PI team. But I'm also going to touch on some work that different members of the various labs have contributed to. And also would like to acknowledge my co-authors on the papers that are already published and papers that are kind of in development. Access to the lands, in which we worked on the projects you're going to hear about are across the Pacific, Northeast Pacific coast. And funders so far have been the Nature Conservancy in particular, and also the Oregon Kelp Alliance and Oregon Sea Grant. So we have a big puzzle of a team, but we fit together really well. And hopefully I can tell you that story in a way that makes you appreciate Pycnopodia. So let's start with a little bit on kelp forests. So kelps are large macroalgae in the order Laminariales. There are other large, functionally similar brown algae, which are sometimes referred to as kelps. But in general, kelps are distributed across temperate, nearshore, coastal zones of the entire world. And kelps are important in particular and known as foundation species. That's because they make foundational habitat and provide food to many organisms. So what is a kelp forest? The last picture I showed shows the typical view a lot of people have of what a kelp forest is, which is just this kind of floating surface canopy forming kelp. But a kelp forest is a diverse place with a lot of components. And we know that, first of all, kelp forests are naturally quite variable and productive ecosystems that are generally threatened by increasing water temperatures. So heat waves are a problem for kelp forests, and also threatened by overgrazing due to ecosystem degradation. And we also know that in general, kelp forests, like I said, because they're known to be variable, it's not uncommon for a healthy kelp forest to boom and bust, to come and go. And especially it kind of depends on the kind of kelp. So there are some that are annuals and some that are perennials. And a lot of times what people don't know about is that there's more than just these surface forming bull kelps. There's stipitate subsurface kelps. And those contribute together to form the kelp forest community. So we're going to go in and do a quick dive at a local kelp forest so you can get a picture of what it's like here in the Oregon coast. So I like to highlight this video because it is a good example of a healthy kelp forest in Oregon that shows a lot of stuff that's going on. First of all, there's a lot of stuff in the water. You see a lot of surge. We see a mysids, so that's these zooplankton are mostly mysids. I've dove all over the Pacific coast and all over the world, and I've never seen mysid swarms like what we have in the Oregon coast. It's quite a productive area known for upwelling, cold water diversity. And so it's not kind of, a lot of times when we're diving and surveying our kelp forests, it's not kind of the scene that you might recognize or see in the kind of majestic cathedral kelp forests that are in Southern California. It's a very busy place with a lot going on in the community. So this is a video showing kind of an outward facing view. When we do our kelp forest surveys now, we also video. And we do a pelagic view like that, but we also do a benthic view. So let's look at a really different type of habitat, which is an urchin barren in Port Orford, also on the Southern Oregon coast. This is the benthic view of our video. You'll see these two lasers here. We have, with our camera, we have fixed lasers that help us measure everything that's in our video. I'll talk about that in a second. But this is a chance to kind of contrast a healthy kelp forest to what we would call an urchin barren. And this urchin barren, that terminology really comes from the fact that all that's really left here is crustose coralline algae, urchin, and then you get a couple other echinoderms and the occasional other species. But a couple of years ago, this exact place that I'm videoing here was a lush kelp forest, pretty similar to the one that I showed at Cape Arago a minute ago. And so this has raised a lot of questions, concerns about what's going on in Oregon's kelp forest generally. And so my team is one of many groups, or several groups in Oregon that have been funded by the Oregon Kelp Alliance, or ORCA, to kind of get a snapshot of what's going on in Oregon's kelp forest right now. So I'm going to touch on that a little bit to set the context for the rest of my talk. And what you can see is that these are our initial preliminary data that have come out of our last year's surveys. And you can see, first of all, two plots that show urchin density on the y-axis with purple urchins and the very different y-axis scale of red urchins. And I want to point out that the urchin barren that you just looked at in Port Orford fits within this box here. But the urchin densities at some of the other places that exist here at the Cape Arago are sometimes higher. And thriving kelp forests can also have comparable or higher purple urchin densities than those in urchin barrens. So these are data from our surveys again, looking at bull kelp density on the top plot, and basically subsurface stipitate kelps that include several species of kelp besides bull kelps. And the point that I'd like to make is that, yes, Port Orford is a great example of a place that we don't have hardly any kelps, but it's a famous urchin barren. But these places where we do have pretty robust, healthy kelp forests also can have numbers of urchins that are potentially alarming if out of context. So I want to make the point that an urchin barren status is based on more than just density. It's a community level designation. And Oregon is missing two of its very important historical predators of sea urchins that might be contributing to the formation of urchin barrens. So what we can do with those kinds of videos, here's a quick example. This follows nicely, I think, on Dr. Randall's talk previously. We can take these stills from our video, and we use our lasers to calibrate the size, and we can do uniform point contact community surveys to understand more than just densities, and actually generate data on the community composition of these habitats. And so, for example, this is the multi-dimensional scaling plot that shows a bunch of community variables all at once into squished down into 2 dimensions, essentially characterizing that different transects are different points on the map on this chart. And you'll see that points that are close together have similar communities. And what this can show is that the urchin barren of Port Orford compared to a kelp forest in Cape Arago are very distinctive in terms of their communities, not so much just the density of one species or another. So why don't I transition now to talking a little bit about Pycnopodia helianthutes, the sunflower sea star. It's a fast, voracious predator. It's world famous. It is the world's largest sea star. It can exceed 1 meter in diameter. Those are pretty rare, but they used to be around. I remember finding them on my dives. They were devastated by the sea star wasting disease, and I'm not actually going to talk much about the wasting disease. This is the one slide where I kind of touch on it. But the point that I'd like to make with this is this is a figure from the IUCN Red List endangered listing. And what I'd like to point out with this is that there are parts of the range where we know that the stars are still sometimes common in Southeast Alaska, parts of British Columbia, but for the most part, they're now absent or very rare, especially throughout the Oregon and Northern California coast. Note Oregon surveys were only 2 that contributed to that report. So there's a dearth of information about what's going on in Oregon. And so for the sake of this conference, this talk, I really want to put, hopefully, what we know about Pycnopodia in context with sea otters. We do know that sea otters and Pycnopodia are complementary in their role of kelp forest support. So, for example, sea otters and Pycnopodia elicit different behaviors in their prey, and therefore different non-consumptive effects. So let's talk about that for a minute. Basically, here's a kind of a conceptual diagram of how these 2 important predators, sea otters and Pycnopodia, can affect some of the players in a kelp forest system that we care about. So I'm going to use the term sea urchins. This could be 1 or 2 different species of sea urchins, all sea urchins, or even you could consider this to refer to any herbivorous species that could be prey for these 2 predators. We know that Pycnopodia and sea otters have consumptive effects, which are net negative for sea urchins. So that's what the negative it is. But because of the fact that sea otters eat like 35% of their body weight every day, or whatever the exact number is, they have a much greater probably direct effect, consumptive effect. And we also know that herbivores have a direct negative consumptive effect on their prey, so kelp forests. But also what's interesting, and a lot of what our research is now about, is the non-consumptive direct effects. So that's represented with this dotted blue line. And we think that Pycnopodia have larger, relatively to sea otters, larger non-consumptive direct effects. So that's like a non-consumptive effect would be a fear response. So we think that Pycnopodia create a landscape of fear because they have a particular chemical cue, smell, that herbivores respond to. And you're going to see some evidence of that in my talk today. So, for example, in kelp forests where sea otters are present, their preferred prey are rarely found out in the open. Large sea urchins can achieve size refuge from Pycnopodia, but not from otters. On the other hand, Pycnopodia can hunt in the cracks for smaller urchins that otters don't even bother with. So they kind of are complementing each other and in both their consumptive and non-consumptive effects. And so this ultimately has this indirect effect when you link the predator to the kelp forest. So we know that sea otters have a very strong indirect positive effect on kelp forests. And what's not known as much is what is that's why I have a question mark here, is like, what are the indirect effects of Pycnopodia on kelp forests in general? So that's kind of a lot of what our research is about. And I'm going to touch on some of that today with two focal research stories about the Pycnopodia predation on purple sea urchins, and also a story about trophic ecology of Pycnopodia in the wild. But I'm also going to cover, without showing any data, mind you, but just some cool storylines about the other research threads from our team and how all these tiles fit together into a complex research program. So you're going to hear about a bunch of different projects that our students and our team are doing. I'll get to that later. So first, I need to review a little bit about the major Northeast Pacific urchin predators. The main predators of sea urchins are humans, sea otters. This is the one picture of sea otters that I have in this talk. California sheephead, spiny lobster, and Pycnopodia. And I'll talk about in a second the range issues with these different major predators. We know there's other predators that certainly eat sea urchins. Some fishes do, but they're not considered to be major predators. The one thing I want to get on to next, though, is that not all urchins are equal in terms of their food value. And that's really important for my first research story. We know that starved urchins in these barrens do not have gonads because they don't have food to eat to build healthy gonad. Gonad is what all the predators want. So humans, we want it, we call it uni. That's the urchin. When you eat urchin, you're eating gonad. And the other predators that we know about that really care about urchins, both sea otters and lobsters, are known to prefer to eat urchins that have more gonad. We actually had no idea prior to the research we did, how Pycnopodia fit into this. We didn't have any data on the amount of urchins that a Pycnopodia can eat and whether they would care if the urchin was well-fed or starved. So that's actually what the first research paper I'm going to talk about focused on. So another piece of context that we have to consider is the ranges of these predators. So this is a map that shows, with this like heavy line and this dotted line, the kind of the distribution of these kelp species, the canopy forming kelps across the Northeast Pacific. And I've kind of just highlighted with this color coding spots along the coast that the various predators that are important for sea urchins are known to be. And note that, I mean, this audience is probably quite aware that sea otters are pretty patchy in space right now. We don't really have any significant populations at all in Oregon or Northern California. And importantly, Pycnopodia was lost through much of its entire range in the sea star wasting event. So we're in an interesting place geographically here where we don't have either of some of the most important predators. And so I'll talk a little bit about the research that we're doing up in Sitka, at Friday Harbor, and at the University of Oregon throughout the course of this talk. So in the first the first chapter really of this story is about this particular question about Pycnopodia and their ability to exert top-down control on nutritionally poor purple sea urchins. So I'd like to highlight one slide that kind of does it all for the methods. After all, I can't tell all of the methodological stories here, but this is lab work that we did at Friday Harbor Labs in 2021. This is Julia Cobelt, who was our project technician. She was key to making all of this happen. And basically what we did is we collected 24 sea stars, Pycnopodia, at reefs in the San Juan Islands. This is the reason we did this stuff at Friday Harbor Labs. Is it's one of those places where if you put in the work, you can get the pycnos. And you can find purple urchins. It's one of the only places in the world that we know of where you can get both of those species still, and do experiments on them. So we basically collected several hundred purple urchins, and we starved half of them, and created basically gonad deficient urchins that are representative of barrens, and we fed the others, creating urchins that were full of gonad and representative of kelp forests. And then we put each of these 24 sea stars through a series of trials. We did a Y-maze trial, where we offered fed and starved sea urchins, or sea urchins and no sea urchins, and looked at Pycno's ability to choose or suss out, smell their prey. And then we also did longer-term, week-long trials where each individual Pycno was in a pretty large tank with ad libitum, like enough purple urchins that they could always eat one. We always replaced them if one was missing over the course of a week. And we did this with fed and starved sea stars. So jumping right into those results, we found, first of all, in these Y-maze trials that Pycnopodia chose urchins over no urchins. So they can definitely smell their prey. We weren't surprised by this, but we had to kind of demonstrate that our Y-maze worked. And moreover, they did not differentiate between fed or starved urchins. So they either can't smell the difference or don't care. So next, I'm going to talk about the results of the predation rate trial, where we learned the numbers that Bob referred to in the introduction, which is that the thing that I want to highlight, though, is that the maximum, what we are characterizing here is possible maximum feeding rates. And so maximum feeding rate is somewhere on the order of about 0.68 urchins per day, or for a given sea star, that's 5 urchins per week. And we found that they, if anything, they actually ate slightly more starved urchins than fed urchins. So this is one of the big, I think, the most important results of our paper. Interestingly, we also determined that Pycnos fed on purple urchins at a higher rate if they're collected in areas with purple urchins. And what's neat about this is that the San Juan Islands has really constrained populations of purple urchins. They're not throughout the San Juans, they're only in certain parts. So we kept track of where we collected our sea stars and where we knew where we collected the purples. And it was just kind of an additional analysis that we threw in, that we're surprised to see that basically it might imply that they become practiced at eating purple urchins. I'll talk about that later as well. So we had each of these 24 animals went through 2 week-long trials where they were fed nothing but fed or starved animals. And we generated many terabytes of video data, and then we wanted to quantify the behaviors of the animals. So we actually, it's quite a challenge computationally and just time-wise for people. So we trained a neural network. And when I say we, this was Julia and Dominic Civitelli, trained a neural network and basically used that. You can see on the right, this is the model running after the network has been trained. And we were able to quantify movements of all the urchins and the sea star through time. And so then we ran all of our videos, all of our, each of these replicate tanks through with this model, and it generated about 5.8 million rows of coordinate data, which Dr. Sarah Gravem, my colleague, was essential for processing. So what we found from all that is then we were able to measure urchin speed from those videos as kind of like a proxy for behavior. And we determined that, for one, starved urchins in the experiments moved faster than fed urchins, which kind of fit with our expectations based on theory that urchins are more brave when they're hungry. We also found that Pycnos themselves didn't change speed when confronted with fed or starved urchins. And so they either can't tell the difference or can't choose more nutritious prey or don't need to. Interestingly, we did find that, on the other hand, Pycnopodia were faster when they were from areas that were without purple sea urchins. So naive Pycnos were more active hunters but they still caught less prey. Again, kind of bringing up this idea that there may be some kind of learning curve, that Pycnopodia and barons may learn to be better hunters. So essentially, we had this model, we had this number that we can expect at the most one Pycnopodia to eat about 5 urchins per week. So we wanted to know, does this matter? What does that mean? So we did a modeling exercise where we modeled the, if we assume predation rates of what we measured in our study, we modeled what the possible urchin densities could be in the field. So this is a picture that shows Pycnopodia density on the y-axis, purple urchin recruitment on the x-axis, and importantly, this heat ramp of possible purple urchin densities. Basically, what this is, the heat ramp is anything in red is going to be a baron, and most of the densities that are in blue are more likely healthy kelp forests. I already mentioned at the beginning of the talk that a healthy kelp forest versus a barren isn't just determined by the density of urchins, okay? But in that context, we mapped what the pre-wasting disease densities were from Oregon, Central California, and Washington, that's these dotted lines, and the densities of Pycnopodia now after the wasting disease. And essentially what we found with this modeling exercise, what we're really changing in the model is what's the predation rate. That's the number that's set for this particular figure of 0.68 a day. And what we found is that back in the day when we still had Pycnopodia before the wasting disease, they can, at feeding rates of 0.68 per day, there's not likely to be any ability of sea urchins, even at really high levels of recruitment, to establish the kinds of populations that exist in urchin barrens. So any model, you're going to do quite a lot, any good model, you're going to do quite a lot of sensitivity analyses. I won't do all of those here, but I'll show you one of the most conservative of our scenarios that we evaluated, where we used the bottom end of the confidence interval on predation rate. And we actually assumed that urchins don't eagerly eat, that Pycnopodia don't eagerly eat purple sea urchins. And still, you see that at the pre-wasting disease densities, our data show that it wouldn't, unless you had very high purple urchin recruitment, there's very low chance that there'd be the ability to develop into urchin barrens with the presence of Pycnopodia. Okay, so a few takeaways from that are that, for one, Pycnopodia do not differentiate between fed or starved urchins. And this is a big deal because this is quite starkly different than what we know about sea otters. Sea otters don't generally like to eat urchins that don't have gonad. It's not worth the time. They're smart. And we don't know if Pycnopodia just don't care or they don't understand, but they just basically go through what they have in front of them and they eat. We found that there was substantial individual variation in the sea stars, and that there are quite a lot of mysteries remaining about the feeding ecology of wild Pycnopodia. So that leads me to another kind of more in-depth study where I'm going to show you about 5 or 6 slides about a different project we're working on, looking at this very question of like, what are wild Pycnopodia eating using a fatty acid biomarker approach. So with this study, we're going to talk about Pycnopodia that were collected at 5 sites in Calvert Island in Canada. And we studied animals from a really wide range of sizes for this study. So not only 5 sites, but stars ranging from about 1 centimeter to much larger, like 0.5 meter in size. And we asked whether the fatty acids as trophic tracers differ among stars in different sizes and places. So to kind of set up what that's all about, I need to break down for you how we use fatty acids as trophic tracers. The idea is kind of cartooned in this conceptual, very simple conceptual food web where you have different algae with different biomarkers, fatty acid biomarkers, that make their way into possible direct herbivores or detritivores in a food web. And then we have our predator, in this case, the Pycnopodia. And we can look at the proportional contribution of the different fatty acids in the predator and try to allocate what proportion of their diet must have come from these different animal sources, or even, excuse me, or even the initial macroalgae that supplied those intermediate consumers. So in order to make sense of that, we have to do studies to understand whether the consumers reflect their diets. And I'm going to talk about that in a second. So first, fatty acids are a wealth of information. They're really interesting molecules that can be used for food web studies. That's because there's about 30 to 70 different fatty acids that we can measure in any given sample. They'll come from several different functional groups that most people have heard of, at least on some level, like saturated fatty acids, monounsaturated fatty acids, polyunsaturated fatty acids. And each of these different functional groups play different roles. The individual molecules play different roles in each organism. So there's so much information that can be pulled out of these kinds of studies. And one of the reasons that you should care is that essential fatty acids are those that are commonly referred to as omega-3 or omega-6 fatty acids. So that's because the last double bond in the molecule is 3 positions back from the methyl end of the molecule. And so that's an omega-3 right here. So these essential fatty acids are only synthesized de novo by algae, whether that be phytoplankton, algae, or plants. And so consumers are oftentimes limited by their fatty acids. And we can process our tissue samples, basically boil these samples down to the fatty acid methyl esters, and we run this through a gas chromatograph, mass spectrometer. And then we get these individual peaks, individual fatty acid molecules that we identify, and then we generate multivariate data plots, which I'm about to show you. So it's a really cool process to dig apart and look at something that's otherwise invisible, and it can tell some really interesting stories. So I'd like to focus just a couple of slides on one of these really cool stories that's being led by my master's student, Aubrey Teradash. And what she did is she did the extractions on the fatty acid signatures of 120 stars, as I mentioned, from several sites in Canada. This is in collaboration with Alison Gaiman as well. So basically what we found is points that are close together. Again, these are different individual stars that have similar fatty acid signatures. So we have all those fatty acid variables running at the same time, and it's a similarity. So we find that, really interestingly, there are certain sites that have similar fatty acid signatures and distinct from the others. Some sites have broader signatures that overlap with the others, as you can see. So Choke Pass and Burke Mid, for example, are very distinct, and so is Mie. But then there's a couple of sites like Quaxua and Mustang that overlap several other sites. And so we're still working on what this means. This is a project that's in prep, kind of new results. But essentially, the fatty acid signatures differ by site. If we test that statistically, that's the clear result. One thing that surprised us, however, is that the fatty acid signatures do not differ between sizes. And again, I mentioned that we had really big and really small stars here. So given what we understood or thought I understood about Pycnopodia are thought to go through ontogenetic changes in diet as they age. And so it's really quite interesting that even very small stars don't seem to have any structural differences in their fatty acids compared to the large stars. This raises many questions we don't know the answer to yet as to why. So that brings up the important context of fatty acid studies, kind of the way it all works. Most fatty acid studies take a consumer, like in this case, cartooned as a crab or with the Pycnopodia, and go, what are the fatty acid proportions in the animal, in the predator? And let's try to make some inferences. And that's what we just did. That's kind of where we're at with the Pycnopodia right now. However, ideally, projects should cover all of these areas. We should be looking at the fatty acid signatures of all the possible prey, and then doing experiments to understand how does a consumer integrate those fatty acids. There's somatic growth, there's conversion due to elongation, desaturation. Certain fatty acids might be catabolized for energy and some egested. And so we have to do studies to measure all that. And in my lab, that's one of the things we do. We do these feeding trials. And so that's actually a plug for the next project that I'm going to talk about, one of the next projects called PicNosh, where we're doing the experiments now on Pycnopodia fed known diets so that we can better integrate our understanding of these patterns. All right, so far, yeah, timing is good. I've just presented two projects that are kind of at like really big extremes in the process of understanding complex ecology of a kelp forest. On the one hand, we have a controlled feeding trial with the sea star, Pycnopodia, and really what we got out of that is really good information about whether they eat starved or fed purple sea urchins, but also kind of a constrained number about their possibility of maximum feeding rates. So one of the big limitations right now is that we don't know, if those feeding rates that we observed in the lab, which are clearly maximum rates, how those would translate in the field. You know, when Pycnopodia have choices of different prey, do they choose urchins? And likewise, do they actually eat that many in the field from what we saw in the lab? So that's one example. We're controlled and focused, and that's the way science works. You have to take one step at a time, and that's the first step that we had to take. It doesn't mean we feel like we're done. And that's why I'm going to highlight a bunch of other things. So on the other end, we have a very kind of first order field trophic biomarker study where we've almost controlled nothing. We just measured the fatty acids of a bunch of animals and gone, let's see what that looks like. Are they different? Both of those studies are valuable steps along the way in our scientific understanding of Pycnopodia. But they're both totally incomplete when you look at the big picture as an ecologist. So that's why I'd like to touch on the happy family of projects that we're working on with Pycnopodia to try to do a better understanding, to gather a better understanding of how it all fits together. So the first one I'll talk about is, and like I said, I'm not going to go into the results from any of these studies. You're just going to see kind of an overview. Is PycnoJuice. And the question with this project is looking at how purple urchin grazing rates may change when exposed to Pycnopodia cues. So the water cue of a Pycnopodia, does it affect the grazing rates? And this is work that my student at the time, who's now Dr. Ross Whippo, did during his PhD. And he had Pycnopodia in a header tank, and we allowed water to drip down into these other tanks where purple urchins were fed known kelp confetti pieces. And we looked at the feeding rates through time. So that's actually a paper that's in revision at Aquatic Biology. So keep your eyes open for that. It's coming and it's exciting. Another project that Ross worked on is looking at this question of prey selection based on a literature review and then initial lab experiments. Because like I said, our first study with purple urchins, we don't really know how that stacks up when they have other options, how those feeding rates might stack up. So one of the things that we thought we would do is start by looking carefully at the literature. There's a huge kind of gap spatially in where the literature is from. There's a bunch of studies in British Columbia and Puget Sound, some in Alaska, some in Central California, and a lot of missing pieces in the middle of California. This largely comes from the 70s and 80s. There was a lot of interest in in Pycnopodia ecology, and people were, doing gut content analysis by sacrificing the animals, or in some cases, gut lavage, and looking at the snapshots of gut contents. So Ross took and looked at what published data were available and started to get it that way. We've also started to do some experiments where we feed, where we have a cafeteria arrangement. So that instead of just one prey available, there's several prey available and the sea star gets to choose. So we're working on that as well. Another kind of version of that is what we call Picnosh. This is an Aubrey Teradash's project. It'll be part of her master's, and where she's doing Pycnopodia feeding rates and looking at biomarkers. So that's work that we did this summer at Sitka Sound Science Center. This is an example of the prey that our 19 pycnos ate this summer. We basically fed them red urchins, green urchins, pinto, abalone, and clams, and dusky turban snails at all ranges of sizes. And so we're looking at their Pycnopodia growth rate while fed these diets, and also their prey selection on size preferences of these different prey. Ultimately, she'll look at fatty acid content, caloric content of the experimental stars and the prey. And so we'll be able to get to more comprehensive understanding of what a fatty acid signature means for a pycno that's in the wild. The next project that I'd like to highlight is called what we're calling Stardust. And this is about whether Pycnopodia cues in the wild, like in a natural reef, reduce grazing of urchins. And so this is work that we have been wanting to do for a while. And Christy Croker was able to say, hey, let's have my students work on this during their winter scientific diving class in Sitka last winter. And so this is being led by the undergrads on the project, Ray, Rose, and Nathan. And this work, essentially what we did is we put a caged Pycnopodia out in a cage on the seafloor, and we put out arrays of kelp at known distances on these lines. And then we let the urchins that were in this barren forage on the kelp if they wished, given that there was a pycno cue trapped in this cage. But we also, of course, had a cage control where there's no pycno. So we looked at the kelp densities before and after deploying these cages. So this is in prep, and a paper is in prep, and this will be presented at WSN 2023 in Monterey by the team. Another project we're calling Star Track, and this is being led by Dr. Sarah Gravem, and it's looking at how active Pycnopodia might affect urchins. So this is kind of cutting. Not many people have seen this video. This is from this summer. So what we do is we experimentally lay out a Pycnopodia, and you can see the diver at time increments lays down a marker that tracks the movement, the track of the sea star through space, through time over the course of a dive. Then at the end of the dive, we collect the Pycnopodia, but we leave the trail in place, the trail of markers. That allows us to go back at 24-hour, 48-hour, and 72-hour intervals after to see how the community has differed compared to a control location where a pycno was not released, but the same type of trail was laid out by divers. You can see the animals fleeing. So that's what we're trying to quantify with this project. The next project that we're working on that I want to highlight is what we're calling Starburst. This is led by Miles Ruff, who's a prospective grad student for all of you out there that might be recruiting students at Oregon State University in the Gravem-Mengi group. And so Miles this summer helped us and is taking the lead on presenting this at WSN this fall. And basically what we did is when we encountered wild Pycnopodia during our fieldwork, we would sample a grid around them at densities and record the densities of herbivores at 0.51, 1.52, 3, and 4 meters out in this kind of starburst pattern. And we're testing the hypothesis that if there's some kind of effect, chemical cue of the Pycnopodia, that there would be a halo like what we've seen in some of our other field work. But of course, this is different because these are natural pycnos that are just existing without us putting them out or disturbing them. So that's the question here. Another exciting piece of the puzzle is what we're calling Pycnocline. And this project is being led by Nikki Sridhar, and she is a PhD student in the Croker lab at UCSC. And what we did here is built several large-scale plumes where we have laminar flow of water, and we have a cage pycno or a free-ranging pycno that can move around in the cage, understanding that there's water moving in one direction. So the smell of the pycnopodia should only be downstream of the pycno at any given time. And what she's looking at is whether the presence of the pycnopodia, whether moving or fixed in place, is affecting the amount of kelp that the sea urchins can eat, and also the behaviors of the sea urchins, as you can see them fleeing the predator cue. So this is work that she started in 2023, and she'll do phase 2 next year. One of the second to the last project I want to focus on is what we're calling pick pens, where we're looking at whether the where pycnopodia in large cages could affect densities of prey and then kelp grazing rates in the field. So this is a never before seen video outside of our group of us deploying our large net cages and deploying pycnos into the cage and testing the net. You can see here at the end of the video that one of the pycnos actually makes it up and climbs over the edge of the net. This is part of our pilot study. So now we have adjusted this approach to have the bird spikes. So we ring the whole net with, there's the pycno. We ring the whole net with those bird spikes that stop birds from sitting on roofs and whatnot. And that has actually kept the pycnos inside of the net. We thought that might happen, but we had to test it to know. So that's coming. We're actually looking for funding to scale this up with mini replicate nets for studies that would start next summer. The last kind of piece I'll talk about is a project that we're calling OnStar, where every time we encounter a pycnopodia during any field work, regardless of where we are. So we have data now from Oregon, Alaska, British Columbia. When we see a pycno, we record basic information about its location, depth, habitat, behaviors, etc. And the plan is, once we have something like, I don't know, a couple thousand observations, we'll do something about it and analyze. We have about 100 observations now. And I am seeking collaborators. So I've had a few different people in different places log pycnopodia for us. So that kind of hopefully provides an overview of what we're doing with pycnopodia. Additional projects are coming. But to kind of wrap it all up, I'd like to kind of bring it back to thinking about pycnopodia and the context with sea otters. So the question that we started with here for this talk is, does pycnopodia contribute to healthy kelp forests? And I think that the evidence we've collected so far, and from what we know from the literature and from what we have from photos like this, and you can see this photo taken by Lynn Lee, was a few years ago. People have long known that urchins flee from the area of pycnopodia. We know that they eat them, urchins. And now we have a little bit more evidence that says, for one, in general, yes, they should be helping contribute to healthy kelp forests. We know that pycnopodia eat purple urchins quickly enough to cause top-down control in a model, and that the pycno declines were large enough that in a modeling context, they would have released top-down control from urchins. And in some of these studies that I touched on from this summer, without giving away all of the results, because those are still coming along, the basic takeaway from those is that the cues that we've been measuring look like pycno cues suppress grazing in the lab and field at about 2 meters away. Moreover, active pycnos seem to cause urchin density reductions from up to 4 meters away in the field for long periods of time, multiple days at times. So, but a lot more work is needed, and that's just the way science works. We do these kind of incremental steps. So I think that to close off, it's key that we consider that pycnopodia are a complement to sea otters, and they both should be helping in the recovery of our kelp forests. And so with that, I'll pause and take any questions that I need to. Thank you. see. I better get my, I'm not sure if I can hear.
Thanks, Aaron.
Sure.
I will second Annabelle with that. It was super fascinating. It's so exciting to hear all the different projects that you have going on right now with Pycnopodia. I really like the footage of the Pycnopodia on the loose climbing over the net. That was pretty funny. But is that happening here in Oregon, those pins?
The pens, not yet. So basically, in order to do that, we need a place that has Pycnopodia. And so Sitka is one of those places. Like we could do those experiments in Friday Harbor would be another place. There's not loads and loads of Pycnopodia in Sitka, but if you know where to look, you can find them if you put in some dive time. So right now, we really rarely see pycnos here in Oregon. I have seen a couple this year on my surveys, but they're extremely rare. So we haven't done it yet in Oregon, but we hope that with time, there are a couple of populations of pycnos that have been reported on here in Oregon, and we're monitoring those. And if we did get to a place where we got a grant and we had a couple of pycnos over the next couple of years, we could do those in Oregon. We'd really like to, because there's different issues in Sitka versus Oregon. So for example, the urchin barrens that we're working in Sitka, they don't have purple urchins. There's very few purple urchins naturally there. So we'd like to do it in Oregon is the short answer. We're just not doing it yet.
Great. Okay, we've got several questions coming in for you. Sammy asks, what's the method for harvesting tissue samples from sea stars for the fatty acid tests?
Yeah, what's really cool about it is that sea stars are famous for being able to drop an arm and still survive. And with the case of pycnos, they've got so many arms, like, over 20 sometimes. So what we do is we just snip the tip of the arm, and they don't like it. They clearly retract, and they don't love it. But I haven't seen any mortality from any of the 50 or so sea stars that we've sampled in that way. Actually, more like 60 now. We just do an arm snip essentially, and cram the tip of the arm into a little Eppendorf tube.
Great, thank you. What does prey on Pycnopodia out in the wild?
Good question. I'm not sure. I know that anecdotally, I'm pretty sure I saw that when you look at those reports of what sea otters eat, every now and then a Pycnopodia will show up. I don't know of anything that really does eat them that much. They definitely eat each other if they're in the lab. Jason Hoden has talked about that being a problem, that cannibalism is an issue, especially when they're really little. So I would expect that, like a lot of things, when they go through this kind of post-larval stage where they're really small and vulnerable to predation by a lot of things. Once they get bigger, I don't know. When they're really small, there's probably a number of things, including other stars, that eat them.
Great, thank you. All right, Jenna asks, I love to see the behavior of this cool species getting more steady, but I'm curious about the contention early in the talk that sea otters have a lesser non-consumptive effect on urchins and Pycnopodia. Has there been any direct testing of this claim? Aren't fluids from conspecifics, i.e. cracked, eaten urchin debris from messy sea otters an important cue for anti-predator behavior? Or has there been characterized, or has that not been characterized in Eastern Pacific urchin species? Sorry, that's multi-layered.
Great question. No, it's a great question. Basically, it was presented as a hypothesis. Like, the scale of those bars are pretty much not understood. So we've done some experiments. So maybe that informed me a little bit, and it's not fair because you didn't know about those experiments. But we did some pilot studies where we had a header tank where we had a pycno cue, and then we had a header tank where we had what we were calling an otter cue. So we would basically crush conspecifics and let the smell of crushed conspecifics. So in other words, non-science jargony, it's like we would crush urchins as if that's a messy sea otter had eaten some urchins and dropped that waste to the seafloor. and let that smell also affect or come into the water. And we didn't see clear effects of the crush conspecifics affecting grazing rates as high as the Q from actual Pycnopodia. But that's a pilot study. We do not have a good handle on that. So basically the idea there is more like logic, that the sea otters are, obviously they float at the surface, they dive down, they grab, and they pull up, and then they eat at the surface. And if there's a queue, it would be a kind of a diffuse scattering of the remains of that prey to the seafloor, but it's not nearly as potent as the chemical smell and cue of a predator that's existing right next to the sea urchins. So basically watching the animals in the field over many dives, you see when the Pycnopodia is near urchins, there's a very clear behavioral response. And however, the response to sea urchins in the presence of sea otters isn't so much a behavioral immediate behavioral response, but like I said, more like they're just not found out in the open anymore. Their behavior is they stay in cracks. And whether that's just because only the ones that stay in cracks are the ones that make it, or whether they're making a choice like, oh, I smell a sea otter, I don't know about that. Hopefully that answers your question, but it's a really good point, and I agree with you that it needs more research.
Thank you. Beth asks, can Pycnopodia be transplanted to Oregon? Are there enough urchins there to sustain them?
They could be, but in general, I think that there's so much going on with this, just in terms of it's a big question, because now we're close, most likely, to having Pycnopodia listed under the ESA, Endangered Species Act. That's being pursued by different people, and so there's probably it's not going to be easy to translocate pycnos from one location to the other. I think the better, however, there might be activities to grow pycnos within within an area, and then release them. I think that from a restoration perspective, I'm really interested in this, but one of the cool things about Pycnopodia is that they're likely to start recovering on their own. We've been seeing them, even though they were mostly wiped out. It's rare, but we're seeing them. And because of their ability to reproduce so fast, unlike sea otters, there's a good chance they can quickly recolonize their previous range much faster than sea otters can, because sea otters have basically a limitation to how many babies they can make per year. And it's almost limitless with Pycnopodia. So I think that translocation into Oregon, into Northern California, is less likely to happen, but it could happen. We just need to do more science to know if it's worth it to really pursue it. And I feel like that's kind of the way we're framing our science right now to our possible funders, is we have to do this fundamental science to know if we should take these more aggressive restoration efforts of translocation or captive rearing and all that.
Wonderful. Okay, I've got a couple more questions for you. Cheryl asks, are there considerations to use next generation sequencing for qPCR for sea star diet studies?
If anybody is doing that, that's possible. I don't know about that. In my lab, I do study trophic ecology using fatty acids and isotopes, but I personally haven't gotten into qPCR. It'd be interesting, but it's kind of outside of my wheelhouse, so I don't know.
Okay. Anna asks, would you theorize that pycnos eating a greater proportion of starved versus fed urchins may have growth or health consequences? And is there any way to test that?
For sure. I do think that while we measured in these, they're longish trials, it's a week, but while we measured that they didn't seem to mind whether they were eating starved or fed urchins, in fact, that they ate more starved urchins than fed, there should be a consequence, in time, over time. And so that could be measured through by continuing to do the kind of studies we did, but then looking at the caloric content, the growth rates, and the reproductive output of sea stars fed those different diets. That takes a lot of work. Be happy to do that, but each of these projects is a huge undertaking that requires graduate students, technicians, and field time. So it's just a matter of finding funding, but I would love to do that work.
Great. Okay, we've hit our time limit. So thank you, Aaron, so much for presenting all this fantastic Pycnopodia work. We're really excited to see the results of all this research. How do people continue to learn more about the research that your team is doing?
Well, I'm on the web. I have a website. We post about that stuff when we can. We go to conferences. So I'd say follow me and my students, and also Dr. Gravem and Dr. Croker, their labs and their students, and you'll hear more about the work. It's funny because the nice thing about highlighting those little projects is that those are going to be a couple years before you start seeing those projects show up in the literature. So that's just the way science works. It's slow, but yeah, that's probably the best way, is just following us on social media, looking at our websites and whatnot.
Perfect. I put your website in the chat, so if anybody wants to find that, it's over there. And we are going to take an hour-long break so you can grab some food for lunch.